Okay, we are going to pick up chapter 14 with slide number 32 and start talking about section 14.3. Okay, so we are going to list the 3 immune components which cause cell lysis in a type 2 hypersensitivity reaction and explain the role of Rh factor in hemolytic disease development and how it's prevented in newborns. Okay, so in this particular type of immune overreaction, we have a reaction that is lysing foreign cells. So basically, um, cells are killed by IgG and IgM antibodies that are directed against the surface antigens of those cells. And typically, that occurs in some kind of a transfusion reaction. But this is also the case in certain types of autoimmunity, where basically the immune system has gotten confused and looks at the surface molecules on our host cells as foreign. And so alloantigens are molecules that differ in the same species but are recognized by the lymphocytes of the recipient. So this is the case where you have something injected like blood into the host that is recognized by the immune system as not self. So this is actually not really an immune dysfunction. The immune system is doing what it's supposed to do, but it's fighting with something that we have decided is a medical necessity, like a blood transfusion or an organ transplant. Okay, so a classic example of that is the Rh factor. With an Rh negative mother, which means that Rh is considered a foreign antigen, and an Rh positive baby, so the baby's blood cells do contain that positive Rh factor. Now, this was first discovered in experiments that were looking for genetic relationships between animals. So rabbits that were injected with the red blood cells of rhesus monkeys produced an antibody that also reacted with human red blood cells. And that monkey antigen seemed to be present in about 85% of the human red blood cells, but was absent in about 15%. And so Rh positive is the dominant gene in the human species, and Rh negative is the recessive gene. And the only way to develop antibodies against this factor is through sensitization across the placenta, and this is the case with the Rh negative mother and the Rh positive baby, or a transfusion that doesn't take this Rh factor into account. Okay, so placental sensitization occurs when an Rh negative mom and an Rh positive fetus have some kind of a blood crossing event. So those fetal red blood cells might leak into the mother's circulation. Often it's during childbirth, or there's a placental abruption where a bit of the placenta kind of pulls away and there's blood that leaks into the mother's body from that placental abruption. The mom's immune system detects that foreign Rh factor on those fetal red blood cells and then becomes sensitized. So this is that first response, or that first immune response. So again, it's going to take a little while. It may not actually attack that first baby. So the first child that the mom has generally is not hurt because it generally happens pretty late in the pregnancy. So the mom's body hasn't actually had enough time to produce an immune response. However, that next pregnancy with an Rh positive fetus, the minute the fetal blood cell escapes and is recognized by the mom's immune system, it's going to have that secondary response. So you're going to have a really big response and a really fast response. And oftentimes it happens earlier in the pregnancy. So those maternal anti-Rh antibodies are generally IgG type, and remember that's the kind that will actually cross the placenta. So it will cross, affix to those fetal red blood cells, and basically lyse the baby's red blood cells. So it will burst open all of the baby's blood, and essentially it is going to kill off the baby's blood cells and ultimately kill the baby if it's not addressed. So the syndrome is the hemolytic disease of the newborn, shortened to HDN, also called blue baby syndrome because all of the red blood cells have been killed off. The more formal name is erythroblastosis fetalis, and it's characterized by very severe anemia as well as jaundice because, again, the baby has produced red blood cells which have been killed, and all of that hemoglobin is still there. And so the hemoglobin is processed down to bilirubin, which is what causes the jaundice. So that's the yellow coloring in the baby's eyes and so on. And of course, because the red blood cells have been killed, the baby is going to be suffering from anemia. And often babies don't survive that if it isn't properly treated. Okay, so again, that first Rh positive interaction generally is a healthy pregnancy because it tends to happen later in the pregnancy. But any subsequent baby is potentially going to be killed by the mom's antibodies if no intervention occurs. So once that sensitization has happened, every other Rh positive fetus is going to be at risk. So any Rh negative mom really has to pay attention to this. The Rh types of all of the children and the Rh status of the father are needed to know if there's a risk. Now, if the dad is Rh negative as well, then you're going to have essentially almost a guaranteed Rh negative baby because when you have two recessive features, then by definition, if you have an Rh negative mom, she has two copies of that Rh negative gene. If you have an Rh negative dad, you have two copies of the Rh negative gene. So the only option is for each of those babies to be Rh negative. But if the dad is Rh positive, then depending on whether he has one or two copies of that Rh positive gene, then some number of those babies that the two would produce are at risk of this situation happening. So there is something called ROGAM, and it is essentially an anti-serum. And what it does is it stops the mom's immune system from responding to the baby's blood cells. It is a passive immunization for an Rh negative mom with an Rh positive fetus. And basically, that immunoglobulin fraction, like other anti-sera, is produced from pooled human serum. And generally speaking, it's injected at about between 28 and 32 weeks of pregnancy. So about 2/3 of the way through. And again, immediately after delivery. And basically what that does is it stops the body from recognizing that Rh negative or that Rh positive blood cell or those blood cells that are released during the birth. So basically, those antibodies, it's referred to as sequestration. They sequester those red blood cells, so they coat it with these foreign or these artificially provided antibodies, and keep the mom's immune system from waking up and having to deal with that. Okay, and it has to be given each and every pregnancy with an Rh positive fetus. Otherwise, the mom's immune system is going to wake up and attack the baby. Okay, so concept check. When is the ROGAM shot needed? Okay, Rh positive mom, Rh positive fetus, Rh positive mom, Rh negative fetus, an Rh negative mom and an Rh negative fetus, an Rh negative mom and an Rh positive fetus. Or all of the choices are correct because mom will always mount an immune response to the baby's blood cells. And the correct answer is D. So Rh factor is something that either you have in your own body and therefore you tolerate the Rh positive antigen, or you don't. If you're negative, that means that you do not have that Rh factor in your own blood, which means that anything that does have that Rh factor is automatically considered foreign. And so it would be an Rh negative mom and an Rh positive fetus. Okay, the next section, we're going to be identifying the commonalities and differences between the type 2 and type 3 hypersensitivities. Okay, so this is a place where you have these immune complexes formed. So it involves the reaction of a soluble antigen. So here we're not talking about cellular antigens. We're talking about something in uh, the fluid, the blood fluid. Um, and it is the soluble antigen and antibodies. And what happens is those antibodies cross-link those soluble antigens and form these complexes, these chunks of antigen antibody complexes. Um, and this involves IgG and IgM antibodies. It also brings in the whole complement system. And unlike type 2 hypersensitivities, the antigens are not attached to the surface of a cell. Okay, so these immune complex reactions produce these free floating chunks of stuff that end up being deposited in tissues. And oftentimes, what ends up happening is that they block certain tissues from being able to do certain things. The kidney is a classic example. So when you get these immune complexes, very often you get kidney damage because they get stuck in the kidneys. Okay, so immune complex disease is where large quantities of these antibodies are produced in response to a large amount of this soluble antigen. Upon the second exposure, again, you get a very fast and a very strong immune response. So basically, you end up with lots of these antigen antibody complexes, and those turn around and recruit a type of white blood cell called neutrophils, as well as the complement system. And normally, the complexes would be eliminated by those neutrophils, but once they get to be a certain size, the neutrophils cannot phagocytose these complexes. And what ends up happening is that these complexes, as I said, get deposited in tissues. So sometimes it's in the basement membrane of epithelial tissue, and it basically breaks down the integrity of the skin. And the neutrophils release these lysosomal granules that actually digest the tissues and just cause a destructive inflammatory condition in the cell. Okay, so here's an example of a skin reaction that you would see when you have this kind of immune response. So this is referred to as an Arthus reaction or serum sickness. And the history behind serum sickness is that this often occurred when antibodies produced by another animal, like you got horse serum to fight off a tetanus infection. Well, tetanus will kill you, so it's better to have this nasty skin disease than to be dead from tetanus. But this was a response to to soluble proteins in that horse serum that our immune system recognized as foreign. Okay, so it's similar to anaphylaxis in that it requires sensitization and preformed antibodies. However, the types of antibodies are different. So in this particular case, it's mediated by IgG, IgM, and IgA, whereas anaphylaxis is mediated by IgE antibodies. In the case of the serum sickness, you generally get a large dose of antigen, and it requires a large dose of antigen to have this huge systemic reaction. And the symptoms tend to be delayed. So it takes a while for the body to build up this immune response. So often it's delayed for hours and even up to days before you see this reaction on the skin. Okay, so which of the following antibody types is not involved in that serum sickness or Arthus reaction that we were just looking at? And the answer is IgE, because that is associated with a more classic allergic reaction, which is IgE mediated. Okay. Section 14.5. So describe one example of a type 4 delayed hypersensitivity reaction and list 4 classes of grafts and how various grafts can cause either host versus graft or graft versus host disease uh, to occur based on the immune reaction. Okay, so a type 4 hyperimmune reaction is a cell-mediated reaction. And so this is involved with T cells. So all the other ones that we've been talking about before have involved antibodies. In this case, we're looking at cytotoxic T cells. So this is results when the T cells are responding to antigens that are displayed on the surface of tissues. So either it is self, but the T cells are confused and they are starting to attack things with self antibodies, or I'm sorry, self antigens, or it is attacking something which is transplanted. So in fact, the immune system is doing the correct thing, but it's not what we want it to do because obviously this is a medical procedure and we don't want the immune system to attack the graft. Traditionally, this is known as a delayed hypersensitivity, and typically symptoms arise between 1 and several days following that second contact with the antigen, or in the case of a graft, it's kind of a continuous sensitivity or a continuous interaction. So a classic example of this is our tuberculosis test. Okay, so basically the tuberculin toxin is injected into the skin, very shallow injection, and the acute skin inflammation that occurs if you have been exposed to tuberculosis is the way that you know that you've had that positive exposure. If you have not been exposed, then you don't have all of the T cells that would normally deal with a tuberculosis infection. And because it's just the toxin, it's not enough to start an autoimmune, or sorry, an immune memory process. And so if you've never been exposed to tuberculosis, you would test negative multiple times getting this test. So this is the diagnostic tool for tuberculosis infections, or to clear people from a tuberculosis infection status. So for example, many healthcare professionals are required as part of their job to get a tuberculosis test, and they have to test negative. That's also true of a lot of aides that work with the elderly. It's true of also aides that work with people with developmental disabilities. They have to test negative for tuberculosis in order to be able to do the job. Okay, so if you are positive, the Th1 cells are going to release cytokines that recruit macrophages and neutrophils, and basically everybody in the immune system jumps on this tuberculin toxin because it's been pre-exposed, and that causes a big red bump. And so that would be a positive test if you had a big red bump. Another type of type 4 hypersensitivity is called contact dermatitis. And this is the case with things like poison ivy, with reactions to certain lotions and fragrances and latex and that sort of thing. So first you have to have some kind of a sensitizing dose for basically your immune system to learn that this is a dangerous thing that it needs to fight off, create the memory, and then once you have a provocation by being exposed a second time, in the same way that the immune system responds harder and faster, it's going to respond that same way to this sensitizing chemical. The allergen, in this case, needs to penetrate the outer skin layers, and then it's going to be processed by dendritic cells that live just underneath the skin, and it's then going to be presented to T cells, okay, which are then, in subsequent exposures, going to attract lymphocytes and macrophages. Okay, and cells are going to release enzymes and cytokines that actually damage the skin in the immediate vicinity. And this is one of the reasons that these poison ivy infections get so angry and weepy and so on, because you've got an enormous hyperreaction of the immune system occurring in that localized area right underneath the skin. So in the case of organ transplantation, this isn't really a mistake by the immune system. This is the correct behavior of the immune system upon finding non-self materials. But again, this is a medical procedure. We do not want the organ to be rejected. Okay? So this organ transplant is a life-giving procedure, but naturally, lymphocytes are going to seek out and destroy any foreign tissue, which this graft is going to be. So the bulk of damage that occurs in graft rejections are those cytotoxic T cells that are killing the tissue that has been grafted in. Okay, so remember in chapter 13, we talked about the major histocompatibility complex, also referred to as the HLA. And this is basically one of the main mechanisms that the immune system uses to know which tissue is self and which is not. And so even though a person can have some variability in the patterns of these markers, in an individual person, it's identical in the similar kinds of tissues in that same person. Okay, so similarity is seen across family members. So the more related a person is, the more likely that a graft from that individual would be accepted by the immune system, because there's less of that non-self milieu of markers to make the immune system upset. So the more distant the relationship, the less likely that those MHC genes will be alike, and therefore the more likely that the organ is to be rejected. Okay, so when that donor tissue displays really different surface markers, the T cells are essentially going to go after it as a foreign substance, which in fact it really is. Okay. So once those T cells recognize that foreign MHC class 1 marker, it's going to release IL-2 as part of a mobilization of the rest of the immune system. So we talked about cytokines being released, and one of the things that it does is it calls other immune cells to it. So those helper and cytotoxic T cells are going to bind to that grafted tissue and start secreting lymphokines that begin that rejection process. So basically, the more immune cells that come in and the more cytokines that are released, the stronger the reaction. And within a certain period of time, it's going to completely damage or destroy that organ. So a lot of the damage that occurs actually ends up being to the vascular supply, which will ultimately lead to the death of the graft. If the tissue doesn't have any blood feeding it, then it's going to die off. The other way is that lymphocytes that come along with that graft might actually be released and turn around and attack the host. So essentially, it's kind of, unlike us identifying foreign tissue in our body, in that case, it is the original owner of that tissue's immune system that's then going to turn around and attack us. That is referred to as graft versus host disease. So the graft will attack any host tissue bearing those foreign, from its perspective, MHC markers, and it becomes a systemic and highly toxic disease. So very often you end up with this peeling skin, as well as other organs being attacked as well. And typically that takes a little longer. So, and mostly because there are fewer of those lymphocytes in the graft as opposed to the rest of our body. And so that's going to occur somewhere between 100 and 300 days after the transplant or the graft. And so here's a picture of the two things that we just talked about. So in this picture, we have host versus graft on the left side and graft versus host on the right side. So host versus graft, you have some kind of a transplant. In this case, we have a kidney, okay? And the kidney cell has an MHC class 1 that is dramatically different than our normal self MHC1. And so the T cell receptor is going to recognize that, and it's going to call in the rest of the immune system to attack that foreign tissue. In the other case, you have a piece of a graft that has those referred to as passenger lymphocytes. Okay, it's going to be looking for an MHC1 that's different than the original donor. And so, of course, every cell in our body is going to have a different MHC1 than what it's looking for. And so it, in turn, is going to attack. Okay, so there are a number of different kinds of grafts that are possible in doing tissue transplantation. So the first one is called an autograft, and that's tissue that's transplanted from one site on a person's body to another. So a lot of times, for example, when you have a burn victim, you will get a skin graft, and they might take a big chunk of skin from maybe the back of the thigh and put it onto the burn with the hope that it will actually take hold and start to grow and become the normal tissue right there. An isograft is one used from an identical twin. Now, most of us don't have identical twins laying around. The few who do are able to utilize that, and normally speaking, it will behave very much like an autograft because genetically speaking, identical twins are going to be the same. Now, the majority of transplants occur as allografts, which is an exchange between genetically different individuals, but from the same species. So, if one person donates a piece of a liver to another person, that would be an allograft. The MHC1 markers are not going to match, and so very typically, you're going to have some kind of immune suppressive drugs that are going to be required so that that piece of liver is not going to be rejected. And a xenograft is a tissue that's exchanged between species. So you might have, a pig liver, and a piece of that would be transplanted into a human. And that one is going to cause an even bigger immune response because not only will the MHC1 molecules not match, but there are going to be a lot of self-surface markers that are not going to look like self to our immune system. So we have had transplants on virtually every major organ, but most commonly it's going to be skin, liver, heart, kidney, coronary artery, cornea, or bone marrow. Those are the most commonly implanted tissues in transplants. And sometimes you can have a live donor, so people can get by with only one kidney. So sometimes family members will donate kidneys to other family members to allow them to not have to stay on dialysis for an extended period of time. And parts of us regenerate. So a liver will completely regenerate after a period of time. So you can safely donate a chunk of liver to somebody else. Your own liver will regenerate, and the piece of liver will form a full liver for the person who receives it. Recently deceased, so if they're able to keep the tissues oxygenated and the blood flowing, then they're able to transplant those organs from recently deceased patients to people who need those organs. And fetal tissue is also sometimes utilized to create organ transplants. Bone marrow transplantation is one of those things. Now that completely regenerates. So if you go in and you scoop some bone marrow out of one person and place it into another person, that will form proper bone marrow in both individuals, and the bone marrow from the donor will completely regenerate. So typically that's used in individuals who have immune deficiencies. So things like aplastic anemia, people who have leukemia. So typically in that case, the person will be irradiated to kill off their own blood stem cells and all of the white blood cells that are causing the problem. And then the bone marrow is transplanted into that individual and starts to regenerate and gives them a functioning immune system. Uh, and people who have radiation damage, they're exposed to large doses of radiation. One of the problems they have is that they no longer have a functioning immune system and can't produce red blood cells. And so by giving them bone marrow, they're able to start producing their own immune system and red blood cells. Um, So again, you try and match the bone marrow as closely as possible to the individual so that you don't have that whole MHC1 mismatch. Okay, and you can still end up with the graft versus host disease, where the bone marrow starts attacking the host cells of the recipient of the bone marrow. So you may, in fact, still need anti-rejection drugs to make the whole thing work. But sometimes when you get that that bone marrow transplant, the recipient actually changes their blood type to the blood type of the donor. So that red blood cell, or I'm sorry, the bone marrow not only implants, but all of the blood cells that it produces kind of look like uh, the, the donor's blood. And so that's kind of an interesting side effect. So interesting way to change your blood type. Okay, so concept check here. Match the class of tissue graft with the description. So the allograft is from a non-related donor within the same species. Okay, so a D would be the right solution for that one. An autograft is from one part of an individual to another part of that same person. So that would be C. An isograft is between identical twins. So that would be A. And a xenograft is the graft between the different species, or B.